Hey everybody, so this is part 2 of the Mendelian Genetics PowerPoint for Biology 160, and this does have some redundant slides from the last PowerPoint, but hey, repetition helps when we're learning new things, so I'm going to go through them, albeit a little more quickly than I will the slides that I haven't covered yet. So let's get started. So in the last PowerPoint, complete dominance was emphasized fairly heavily because that's kind of a good starting point. It's easy to understand. One thing swamps out another. But there are other styles of allele that exist in the world, so we need to make sure that we address those as well. So in incomplete dominance, instead of having an F1 generation that is identical in phenotype to the homozygous dominant parent, you get a mixed phenotype, so a mixture of red and white in this case, or a mixture of whatever phenotypic outcomes the two alleles of a particular gene have. So heterozygous individuals show a blended phenotype, whereas homozygous individuals show a non-blended trait. So what's significant about this is that if you allow the F1 generation to self-pollinate or self-cross, so cross a heterozygote with a heterozygote, the F2 generation has not only a 1 to 2 to 1 ratio of homozygous dominant, homozygous recessive, and 2 heterozygotes, so 1 to 2 to 1 with the genotypic ratio, but the phenotypic ratio matches. So 1 to 2 to 1 red to pink to white. So if this were a complete dominance scenario, you wouldn't see the same phenotypic ratio as genotypic ratio. So if that was the case, and red flowers always swamped out any whiteness at all, you would see a red F1 generation, and you would see a 3 to 1 red to white phenotypic ratio. So you can actually tell whether a trait is completely or incompletely dominant by paying attention to the outcomes of particular kinds of crosses. Oops. All right, sometimes things are codominant as well. So what this means is that there are no recessive alleles. All alleles are equally dominant in this case, and if there's more than one kind of allele present, both will be expressed. So the best example of this in this slide is the AB blood type. So this person has one copy of the allele for type A and one copy of the allele for type B on separate homologous chromosomes, because we want to be mindful of using our terminology correctly. But because they both exist and are equally dominant, both are expressed on the heterozygote's red blood cells. And I want to stress here that O, it's not the case that this red blood cell is covered with type O glycocalyx elements, but rather it just doesn't have them at all. So that's what type O means. It's not the presence of an O allele, it's just an absence entirely. Okay, so we've been talking about traits that are controlled by one gene with a couple alleles. Some traits are controlled by multiple, and that means 3 or more, so more than 2, alleles of the same gene. So blood type is an example of that. You can have A, B, or O, so there's more than 2 kinds. But each individual can only have 2, and that's because you can only have 1 per homologous chromosome. So you would never see an individual whose blood type was A, B, and O. That's not possible due to the way that our chromosomes work. So most traits are controlled by multiple genes. So within each gene, there's possibly more than one allele of the gene, but the trait itself is controlled by more than one kind of gene. So for these, you tend to see a continuous distribution rather than phenotypic ratios. So instead of saying like 1 to 2 to 1 ratio of short to medium to tall people, you see a continuum where people are on either the shorter end of the spectrum or the taller end of the spectrum. Same with eye color and hair color. So they're all controlled by multiple genes, which is where you get a lot of possibility for variation. Okay, so now we're getting into X-linked inheritance, and notice it's X-linked, not sex-linked, which begs the question, well, why X in particular? What's special about the X chromosome versus the Y chromosome? Well, if you look at the relative size of these things, and you'll see a picture of this in a minute, but why not draw it right now? The X is a pretty big chromosome compared with the Y. Remember, the Y doesn't really look like a Y, but we're just going to illustrate it that way anyway. So what that means is that there are more genes on the X chromosome than there are in the Y chromosome. So this one has fewer genes. So if an individual inherits an X chromosome that has broken or harmful allele on the X chromosome, and they don't have another X chromosome with a good copy to compensate, which is to say they have a Y, not an X, then they're going to manifest the condition, whereas a female individual with one bad and one good copy wouldn't, but they would be a carrier. So that's kind of the way this works. So certain human conditions, and here's just a couple examples. So we have red-green color blindness, which is far more common in males than in females, and hemophilia are called X-linked because they stem from an allele that is dysfunctional and that is located on the X chromosome in particular. So here is the X and Y chromosome respectively. You can see that the Y chromosome is very diminished compared with the X. So not only is it gene poor, but it's really, really little. It's very small. All right, so with X-linked inheritance, men are more likely than women to suffer from these conditions because men only have a single X chromosome. So they don't have two copies of X where one good copy of a gene could be on the other chromosome. So if a woman is affected, she needs to have two recessive alleles, one from each parent, to be affected. So it is possible for women to be affected by these disorders. It's just not nearly as likely. So that's what we're seeing here. So let's actually go to the whiteboard for a minute and game this out. So I'm going to use the power of Epic Pen, which I love so much, to make a Punnett square or a Punnett rectangle, as it were. So let's do some sex chromosome stuff here. So let's say that this is a normal X chromosome, and this is an X chromosome, and I'm going to pick a different color, and we're going to just give it a little asterisk right here. So that one is the one with the allele that is involved in hemophilia. So let's cross a carrier female with an affected male. So we have, in the top row, two possible daughters. And I'm saying possible because remember, these are probabilities, right? So this is just a chance rather than this couple will have 4 children and one of them will be affected. All right, and then let us go down here and we can do XY, XY. So as you can see, there is a 50% chance of having an affected child. There is only a 25% chance of having an affected son. And there is a 25% chance as well of having an affected daughter. So that's kind of the breakdown, and I expect you to be able to do this kind of thing if I give you a Punnett square or I give you a scenario problem. Be able to break down the probability of different phenotypic outcomes from Punnett squares. So this is an example using X-linked traits, and there are, of course, others. But for now, let's move on. Okay, so we've established that. Whoopsie. Here we go. So if you look at this picture and you see either a 3 or an 8, you are not red-green colorblind. But if you just see a bunch of dots, you are. So this is a test that physicians and clinicians can use to test for red-green colorblindness. Okay, so let's talk about dominant versus recessive genetic disorders. And these are going to be primarily autosomal. So the autosomes are chromosomes that are not sex chromosomes. So most of them. So a recessive disorder is what it sounds like. A recessive gene is masked by a dominant gene, but homozygotes for a recessive genetic disorder express the problem, whatever it is. So you need 2 copies of the recessive allele in order to have the disorder or disease. So heterozygotes don't express the disease, but they do carry it. So one example of a recessive disorder is sickle cell anemia. So this is a disease that affects the gene that codes for hemoglobin. And a change in the way hemoglobin is synthesized results in a concomitant change in red blood cell shape. So this cell right here is one of the sickle cells, and you can see another one poking into the frame right here. Okay, so autosomal disorders happen on autosomes. So if there's an autosomal recessive disorder, that means that persons must be homozygous for the recessive allele in order to suffer from the condition. So for example, sickle cell anemia, they need to get 1 copy of the recessive allele from mom and 1 copy of a recessive allele from dad. So if, let's make it red because it's about blood. Large S equals normal, little s equals either a carrier or actual sickle cell. I'm going to do this in this color with Epic Pen so it stops doing this spiky thing. So only individuals who are little s are affected. Someone who is big S, little s is a carrier, but unaffected. So that's the deal with that. So if I tell you that an autosomal genetic disorder in a test question is a recessive disorder, you should be able to predict the outcome or the chance of a child having the disorder using a Punnett square. And you should arm yourself with the knowledge that you need 2 copies of the recessive deleterious allele rather than just 1. But that's not always the case. Sorry, I'm just going back and erasing what I wrote. There we go. Okay. So carriers are heterozygous for the condition, and the alleles they have for the trait in this case differ. So one is functional and the other is dysfunctional. But because this is a recessive disorder, the dominant allele is preferentially expressed and protects them from experiencing the condition themselves. So they just carry it and can pass the disease to offspring. So you might be wondering, okay, well, if sickle cell can kill you, and it can, why would it persist in a population? So now that you know about evolution and how natural selection works, you might assume that a disease like sickle cell would end up being selected against and therefore get purged from populations experiencing it for that reason. But the reason that it seems to have persisted is because in areas where people have sickle cell alleles, and there are malaria-carrying mosquitoes, people with sickle cell have resistance to malaria. So An individual with sickle cell may suffer some health problems related to their sickle cell. However, if there's a malaria outbreak in their community, they're a little bit more likely to survive than someone who doesn't have sickle cell anemia. So that is the current school of thought for why this disorder has stuck around. Okay, so dominant genetic disorders. A single allele, the dominant one in this case, can bring about the condition, regardless of whether a person also has a normal allele. So if you have even one copy of the bad gene, you're going to manifest the condition. So in this case, we have Huntington's disease, which is a neurodegenerative disease that strikes in middle to later age. So people with Huntington's experience loss of neurons and therefore loss of brain tissue. They experience dementia, and they experience uncontrolled movements. And this is a disease that is not curable. So once the disease begins, symptoms can be treated, but the prognosis is ultimately terminal, which is very sad. So understanding how this disease is inherited can help us as a society avoid propagating the negative allele. So let's compare and contrast two Punnett squares. In the top one, sickle cell anemia, which is a recessive disorder, there's a 1 in 4 chance or 1 in 4 probability of inheriting the disorder if you cross two heterozygotes. So we have a 1 to 2 to 1 genotypic ratio, but a 3 to 1 phenotypic ratio. In the case of Huntington's, if there is an affected father, but mom is not affected, there's a 50% probability of individuals passing the disorder along. And that's a big chance, you know, that's 1 in 2 children. So this is something that couples really need to consider if they have a history of Huntington disease in their family. Now, I just listened to a really amazing episode of This American Life concerning a doctor who, well, I'll let you listen to it. It ends up, well, the story ends up with a Huntington disease diagnosis, but the ride to that diagnosis is pretty wild. So it's this, which is a podcast and also a radio show. And the episode, I don't remember the exact title. It's like Dr. No, not Glimmer. Haha. Gilmer and Mr. Hyde. It's very, very interesting. So if you want to engage in some relaxing science listening with a touch of true crime over your break, you can watch that. Alrighty. So, aneuploidy. So we've talked about some forms of ploidy before. So diploid means 1 copy from mom and 1 copy from dad. Haploid means 50% of the genetic information of a normal cell. So these are gametes, and these are somatic cells, so body cells. Aneuploid is when there's just an improper number. So not a doubling or halving or tripling, but rather 1 chromosome is lost or gained within one of those pairs. So that's why we get usually 45 or 47. So you usually lose 1 because we should have 46. 46 chromosomes total, 23 pairs. But if you lose 1, you get 45, and if you gain 1, you get 47. So these are caused by error in meiosis, specifically non-disjunction. So 90% of the time, in cases of Down syndrome, so this is an extra chromosome number 21, it is non-disjunction in the egg, and that is because the proto-eggs in each ovary are all of the eggs that will ever be available to be ovulated by a woman. So the stem cells to make more of those pre-eggs go away prior to birth. So each egg that is ovulated represents one chance for fertilization, and once you're out of those, you're out of chances for fertilization. But that means that as a woman ages, the eggs that are left later in life have had more time to accumulate mutations due to, let's say, a woman had a lot of x-rays or lives in a heavily polluted area where there's lots of teratogens, which are tumor and mutation causing agents, a variety of things. So after 35, a woman's chance of having a baby with Down syndrome goes up dramatically. And this is actually the reason for a funny phenomenon in medicine. Geriatric medicine is the study of treating old people and keeping them healthy. Having a baby after age 35, they characterize as a quote-unquote geriatric pregnancy, which many women take issue with for obvious reasons. So women nowadays are waiting later and later to have children to get career goals down first. And so to have your OBGYN call you geriatric is pretty offensive, and many women, as you might imagine, don't like that. So we talked about Down syndrome, which is an abnormal number of chromosomes at position number 21. And the reason that that is survivable is because the higher you get in chromosome pair numbers, so leading up to 23, which is the sex chromosome pair, the smaller the chromosomes get, meaning they contain fewer genes. So having an extra one or missing one is not as big of a deal. Turner syndrome results in sterile phenotypic females with no second X chromosomes. We call them XO. But you need at least one. So the minimum criteria for sex chromosomes is you gotta have an X. And that's because we know from studies, if you don't have at least one, pregnancy isn't viable, so you get spontaneous miscarriage. Because there's some important stuff on the X chromosome, stuff you need. So Klinefelter syndrome is the opposite scenario. So this is genetic males with an extra X. And I want to stress here that it's not totally correct to call them men, but rather males. So males is the name that science has awarded to human heterozygotes at the sex chromosome position. But there's plenty of people with Klinefelter syndrome who were raised as women and who consider themselves to be women. So this is a little bit of a error. So let's cross that out. Okay, so moving right along. Aneuploidy can come around in regular cell division as well as in meiosis, but usually it happens due to the DNA that controls disjunction and cell division getting damaged, and then the outcome cells are more likely to become cancerous. So that's why if you do a karyotype of cancer cells, typically you see different aneuploid cells, which is bad news. Okay, so finally, we need to talk about changes to gene expression that are not intrinsic to DNA. So what I mean is, genes can be turned on or off, or they can be enhanced or reduced without actually altering the content of the genome, but rather by making them harder or easier for polymerases to access. So there's two ways we can do this. One is you can wrap the DNA really tightly around the histones. Remember, this thing is called a histone and is made of proteins. So just as an aside, since you have an exam coming up, this is a good piece of information for me to demonstrate a principle about the exam. So what it looks like to have deep knowledge and mastery of something. So things about histones that you need to know. Type of macromolecule is a protein. That's good. That's not good enough by itself because it doesn't tell you what its purpose or its function is. So its function is to stabilize chromatin. So it provides something for DNA to wrap around. And in this particular context, the tightness of wrapping is playing a role in epigenetic gene expression changes. So that gives you the context under which it performs certain tasks. So that would be deep knowledge of histones. So you should be striving to achieve that level of knowledge for all of the terms that have been emphasized in this class, because really the way biology works, everything is related to everything else. You don't ever have the luxury of learning a fact and then forgetting it, because all of this stuff keeps coming back in various forms. Okay, so epigenetic tags are chemical markers on DNA. Frequently, the way that genes can be altered or turned off in this case is by adding a methyl group. So here's some chemistry again. So if I, on the test, I'm just going to move this real quick. Oopsie. No, be bigger. If I, on the test, ask you what of the following chemical compounds disables or is used to tag DNA, and I give you this is a carboxyl group, this is an amino group, CH3, that's a methyl group. So many, many, many compounds have methyl groups. So they're not limited to epigenetic functions, but it is one application of them. So you can use them to block regions of DNA that you do not want to have transcribed and translated. Turns out also epigenetic changes to the genome, oopsie, are heritable. So it used to be thought that during meiosis, some magical process wiped off all of the epigenetic markers that are changing gene expression, but it turns out that just isn't true. So evidence that some markers are not erased and can be passed down from parent to child in the gametes basically is mounting. So specifically genes that involve how individuals metabolically deal with stressful situations, those markers are heritable. And so it's beginning to be understood that these could be responsible for many human conditions that were previously less well understood. So we knew that people developed them, but not necessarily why. It seemed to be random chance, but it might be the case that that's not really true. Okay. So this has unlocked an entirely new field in medicine, and you can understand patterns of behavior and physiology based on epigenetic markers. So we're actually not going to do the take-home assignment because too late in the game for that, and you guys have enough on your plates anyway. But this is like the cutting, bleeding edge of science right now. It's a really exciting field, so it's something that you could potentially get a master's or PhD in if you were so inclined. Okay, so I believe this is the end. Let's check. Yep, sure is. Alrighty, I'm going to post this right now, so thank you for your attention, and I will see you in the next video.